I got to speak up. Wonderful disease we have. It's easily treated and easily diagnosed, and we have a room full of people every time we do something. I'm not going to slam anybody. My wife won't let me. Just wanted to say thanks for coming. About 5-6 years ago, I was told I had chronic fatigue, fibromyalgia, beginning stages of MS and leukemia, and none of it was true. Since then, I met a gentleman, Chris Montes. I gave him a call, talked to him on the phone for about 45 minutes, and he said he'd bet his next 3 months' salary that I had Lyme disease. And that's where we're all headed. So I'll let Chris take over. Thanks for coming.
You never gave me my money back either. Welcome, everyone. I'm so pleased that you're here. This is absolutely phenomenal. There are many, many people to thank, and I know I'm going to leave many of them out, but first and foremost, I want to let you know that Randy and Laura Sykes have done the lion's share of putting this together, so let's please give them a big thank you. Thank you both, as well as others from the Greater Hartford Lyme Disease Support and Action Group. That's to name a few names: Ann Schwing, Steve Arndt, Ruth Kubik, and others. The town of Farmington, Nancy Parent. I know I'm missing people. Thank you so much. You've all been wonderful, very wonderful. And I want to also recognize the representatives from states and even countries who are here. We have people from Canada here, Maine, New Hampshire, Vermont, Massachusetts, of course, Connecticut, Rhode Island, New York, Pennsylvania, Maryland, Florida, New Jersey, Delaware, North Carolina, California. Anywhere else? Virginia. My goodness. God bless you for being here. We have a very full agenda today. We did start a little bit late. We know this place isn't the easiest to find, so we let people come in a little bit later, and also we had to get some technical difficulties taken care of. But I do want to get things going here. I know you've come a long way, and you're excited to hear and see what there is to be heard and seen. So I would like to introduce our Master of Ceremonies. She is a rheumatologist practicing in the state of New Jersey. She has a master's degree in public health as well as her MD from Witwatersrand Medical School of South Africa. She's been working in the Lyme field for a number of years and has been involved in legislation in the state of New Jersey. She's had many, many public speaking roles throughout the United States. And she's here today to be our MC for the rest of the day. Please welcome Dr. Leslie Ann Fine.
Hi, thank you. My degree wasn't actually from WITS, but that's okay. It's all in this green handout has everybody's credentials and their CVs, and I'm not going to read each one. You should look at them because we have the most extraordinary group of speakers here today. I know I'm interested in this field because of the obvious relationship between infections and autoimmune diseases, and for some reason I find that fascinating. And we have managed to get a group of people with the same interests, and this is the first conference we've ever had where it has not just been devoted towards Lyme. The theme here is the relationship between multiple chronic infections and autoimmune diseases, and even a little bit step ahead of that, is there such a thing as an autoimmune disease, or is it all or largely a response to an underlying infection? I'm incredibly excited that we've got these speakers. These are outstanding researchers, brilliant minds, and not concerned about going against the current tide or current medical opinion. Our first speaker is Joanne Whitaker. Again, so many credentials that I'm going to refer you to the booklet. She's well known, she's incredibly brave, incredibly smart, and I'd like to introduce Dr. Joanne Whitaker.
Thank you very much. I would like, before I start to speak, I would like to say that the most important thing of the entire conference is that Randy and his group have gotten together a bunch of right-minded. I'm into whole brain thinking, but anyway, the whole deal is that everybody is getting on the right page, and we better get on the right page, because this is the smartest bug I have ever worked with, and to put up with all the crap that goes with it is a little bit too much. So I think that most of the people in this room, I'm a very sensitive person, and I feel the good aura, and it's wonderful. I haven't been around anything except negativity in so long, my body doesn't quite know how to respond. So if I do a few things crazy, it'll just be by nature. I could blame it on my Lyme disease because it causes most everything. And I've had it since I was a little girl, and it's been called everything. When I found out that there was no good test for Lyme disease, man, I got to work. Now, I want to thank all of you, and especially Randy and all his helpers. 1 month ago, I had a stroke. I could not move anything on my left side, and I couldn't talk. You gave me the goal to come here and talk with you. And man, I can talk now. First slide, please. Now, I have many diseases that I could talk about, but I'm going to talk about this one first. And I want us to all answer this question today by, I hope you can after the information I give you. Is amyotrophic lateral sclerosis, took me a long time to get that out at first, but anyway, blah, 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 blah. ALS is easier, the last stage of Lyme disease. And I want to dedicate this talk to Eleanor G. Fort and my staff, because we wouldn't have any data without them. And Eleanor G. Fort, my friend of 50 years, was with me all over the world, just died last month. She had Lyme disease, but she also had cancer, and this is occurring more and more all the time. And there's a man that's treating the cancer just by treating Lyme disease, and even the metastases are going away. Now, this is important. This makes me have the goosebumps. I used to be a pediatric hematologist, oncologist, and all I did for 40 years was watch children die. So, next slide, please. Now, I've had a lot of experience with the fluorescent antibody technique, and I've used it in many different situation. When I was an oncology fellow at Sloan Kettering in New York, I learned it, but I used it as a tumor marker. Next slide, please. Oh, I then went to the Children's Hospital, and I was learning about all the things about the blood. And all the babies were dying in the nurseries, and nobody knew why. So I whipped out my fluorescent antibody technique, and I found that 2 nurses actually were carrying pathogenic E. coli. So we cleaned up the 2 nurses and cleaned up everything, opened the nursery back again, and we had no more trouble because we used the fluorescent antibody technique to take stool specimens on everybody that came in. No more babies died, but a whole lot of babies died before we used the fluorescent antibody technique. Next slide, please. At Southwestern Medical School, I developed the rapid identification of many infectious diseases, which was very helpful. We used it right in the clinic. We worked on syphilis, pertussis, diphtheria, and I also did the toxin as well as the bacteria, which was very helpful. We took a throat culture on anybody that had a sore throat, and if they had beta hemolytic strep, we treated them with antibiotics. If they didn't, we didn't, and we could have the test rapidly, immediately, so that the patient was treated before they left home. Next slide, please. In 1996, I finally found out that I had Lyme disease, but I had to create my own test to find out. But anyway, I really started to work on when I found out the tests weren't any good. And so I've used this basic fluorescent antibody technique of the experience I had before, so it was pretty easy to adapt it to Borrelia burgdorferi, the causative agent of Lyme disease. And then we went on to develop a quantitative test because we thought this might help in treatment. Next slide, please. This technique is a specific fluorescent antibody technique to identify Borrelia burgdorferi, the causative agent of Lyme disease. The important thing about it, it identifies the antigen, the bacteria, rather than the antibody, and that's the reason it works better than anything else. It's supposed to be a very accurate test for identifying Borrelia. And the results are available and pictures back to the physician within 24 to 48 hours. That's mailing it back. Next slide, please. It is a patented test since January 4th of this year. And anybody doing this test that has not bought a license from me is in jeopardy and breaking the law. They can buy a license, and I'll be glad to teach you how to do it. I'd like to get out of the business. I got more fun things to do in my late life. Next slide, please. Now, this probably is the most important slide of the whole thing, because during this period of time, we had these bloods sent in to us, and this is what we found. We had 195 patients that were clinically diagnosed with ALS, 317 MS, 131 Alzheimer's, 85 lupus, 2,122 chronic fatigue, 1,666 fibromyalgia, and to me, fibromyalgia is just a descriptive term for Lyme disease, and 323 Bell's palsy. All of these had Lyme disease, and probably none of them were treated properly. Next slide, please. Now, amyotrophic lateral sclerosis, ALS, is actually a pathological diagnosis. If you break down the word, A means without, myo is muscle, tropic means to turn, and lateral sclerosis is where it occurs in the brain and spinal cord. So it really means muscle without turning occurring in the brain and spinal cord. Next slide, please. It was first described in 1874 by a French neurologist, Jean Charcot, and he characterized it as an upper and lower motor neuron disease, progressive neurodegenerative disease. Now, what is interesting to me is the cause and the cure remains unknown despite vigorous research efforts. Now, it also just absolutely blows my mind why, and I think I figured out, why anybody would diagnose amyotrophic lateral sclerosis rather than Lyme disease, since there's no cause and there's no cure, and there is a cause and a cure for Lyme disease. Next slide, please. What we did was we took patients' records that were sent in to us as ALS, and we had them, we're doing research on all of our patients, so we had them fill out a specific questionnaire each time they send in bloods and many other important research activities. The Borrelia burgdorferi, the causative agent of Lyme disease, was identified using the Q-rib test, the quantitative rapid identification of Borrelia burgdorferi test, with whole blood and spinal fluid. I found it in everything. I found it in placenta, everything. And Dr. Mattman, who's sitting in the audience, that wonderful lady who knows more than anybody, and I believe this too, Lyde, I'm not just saying it. So anyway, she has identified these bugs too by culture. The next slide, please. Now, when we see our positive test, what we see is a bright green fluorescence, and then we can look at under phase microscope. Would you go back, please?
We're experiencing technical difficulties. Please stand by.
Stand by, everybody. I've experienced a lot of these in my life, so I can handle it. That's when we see the green brightly fluorescing. Then we go to identify exactly what the bacteria looks like, and we can identify it under phase or regular light. I don't know, auto in progress, does that just mean I just keep talking? Thank God I can do that now. A month ago I wasn't able to. I guess some of my friends kind of liked it, though. But what we do is we do a serial dilution on these bloods, and the lowest serial dilution is 1 to 2, and the highest serial dilution is 1 to 128. My dog, Jazz, and I both had 100, the highest you can have, 1 to 128, and I have continued to vacillate back and forth with this. I'll keep talking, or you? So anyway, whenever I go off my antibiotics, I go straight back up to 128. I'm going to do the Bowen technique, that's what I do most of the time. And it's a hands-on technique that balances the autonomic nervous system. And it is fantastic, and I have been doing that on myself all the time too. So we're going to be doing the Bowen technique, we're going to have nutritional counseling, and we have a special exercise program made by a friend of mine who's the international Tai Chi master of the world. So that's what our clinics are going to be about. We're going to be about preventive medicine and preventing disease, not just treating dis-ease. And I've even taught the Secretary of Health in the state of Florida into this too, so that's the big clap should come. Now, we thought that maybe the serial dilution, that the higher serial dilution would be seen in these sicker patients, but that doesn't seem to be true. If you have one bug, I think you got a whole bunch of bugs and it's just not showing up. In fact, sometimes when we're treating the patient, our quantitation goes higher, because I think what is happening is that the bugs come out of the brain and the heart, which they love best, and go out into the blood, and then the antibiotics and the rest of the treatment can get to them. Now, as all of you know, it is not unusual to have other co-infections with Lyme disease. Now, these people, these 104 patients with Lyme disease, 85 of them had no co-infections. 16 of them had Babesia, Now, the geographical location for these 104 patients, there were 7 different countries. We get bloods for our test from 14 different countries outside the United States. And I'm ashamed to say that many times we get more bloods from outside the United States than inside the United States. Now, something is very wrong with this. Now, think about it. Sometimes the Lyme disease helps you. One thing it has helped me with, my patience is a little bit better, not too much, so. Now, the age of these patients ranged from 26 to 78. And the gender, you know, a lot of people say, oh, women don't get ALS. Well, they sure do. They just don't, they won't pay attention to them because they think they're just giving them stuff. Well, it's real in women, too. So there were 70 males and 34 females in our study. The duration of the illness, when these people had their Q-rib test, they had been sick, all of them, for at least 6 months to more than 3 years. So these people were sickies. Well, you don't get diagnosed with ALS. That's the main reason people get diagnosed with ALS. They're sick, and all the doctors tell them, well, you're going to die in a certain period of time. That's lovely, right? Needless to say, I'm a little off on the doctors, because what happens in my case is many of them will not send the bloods to me to get properly diagnosed for Lyme disease. If they do send the bloods to them and it's positive, then they won't treat them. And then if they do treat them, then they don't treat them long enough and holistically. We've got a long way to go. So I'm going to start working with the vets. I'm going to do a test for, since I found many animals have this disease. Maybe somebody will pay attention to the animals. They don't seem to pay attention to the humans. So maybe the vets will listen, and they will. I found the vets will listen. So we'll see. I'm going to start treating just animals in a new clinic that I'm started in Winter Haven, Florida, which is very close to where I am. Now, the whole deal is how many of these 104 patients have been on antibiotics. Now, I'll show you on a slide. You really can't trust what the patient says either, because they put down, please put down all your antibiotics that you've been on or are on. They put down Celebrax and all kind of different things. So anybody knows that's not an antibiotic, but anyway. So you really don't know when they tell you if they've been on antibiotics, if they really have been on antibiotics or not. But anyway, the 73 of the 104 reported that they had not been on any antibiotics. Oh, you get to see it all over again. I'm about through, but anyway. 30 patients say they were currently on antibiotics, but unknown dosage and unknown duration. So I don't really think you can trust any of these things. And it is very difficult for me even to stay on the antibiotics, because you have to eat, number one, and I have no appetite. So I'll eat to take my antibiotics. Now that's pretty awful, isn't it? But anyway. Okay. I just want to go over these because these people have a lot of symptoms and a lot of them were dead. I did a follow-up by sending them the questionnaire again that they filled out before, and then also I did a follow-up phone conversation with them. Okay, so here these are the general symptoms for the 104 people that we did the Q-rib testing on. And y'all can read better than I can, so we'll just continue. Next. These were the neurological symptoms reported by these 104 patients. A lot of them reported weakness And balance is the hardest thing I have. That's one reason I'm in a wheelchair and use my cane all the time, because I'm afraid of falling. Paralysis is very common. Tremors is common, and stiff neck, that just means that you have got some neurological something going on. Next slide, please. Now, these are the mental signs and symptoms. I think everybody that has Lyme disease, or that I know of, they call it brain fog, and we said that we had only 3 that had brain fog, but that should go up into memory loss, because there's nothing more frustrating than being given a talk and coming to a word that you know very well. I know if y'all all have Lyme disease, you know what I'm talking about. But when you talk with people who haven't had Lyme disease, they think you're crazy, and that's the reason. More psychiatrists diagnose Lyme disease than anybody else, because the regular doctors just get tired of messing with them. So they show them off to the psychiatrists, and I've been training them, so they're doing a good job of getting bloods to me. Next slide, please. But the most common signs and symptoms you can see. And speech difficulty was one, but that came from my stroke. But fortunately, I have no residual, and you know I can talk all right. Next slide, please. Now, as I said, we set follow-up questionnaires to these people, and it was from a little bit of time to a long period of time. And only 28 of the 104 questionnaires were returned. And then I made follow-up phone calls on them, and 8 of them had already died. Next slide, please. And the 8 who had died, the questionnaire follow-up, 8 were dead. 11 reported they had an increase in severity of their symptoms. 9 reported less severity in symptoms, whatever this means, I don't know. Next slide, please. And this again is questionable. These were the 8 that died, and it was from 2 to 8 months until death. 2 of them said they were on antibiotics prior to testing it, and 5 of them said that they were on post. But we do know the duration of their illness was from 8 months to 3 years. This is a wicked disease, and this is the reason I think it's the last stage of Lyme disease. Next slide, please. Now, as I talked with you about, the serial dilutions didn't seem to make much difference, but most of them did seem to have a serial dilution of higher than 1 to 16. Next slide, please. And the geographical distribution, they were 5 different countries and 16 from the United States. So you can look up there and see if there were any in your state. And I don't think it has anything to do where the disease is located. What I think it has to do with is somebody's ambitious enough to send the blood to the right place to have it diagnosed properly. Next slide, please. These are different countries and states. Next slide, please. And the age group was very similar to the 104, 27 to 66, and the gender was about the same too, 20 and 8. Next slide, please. As I said, most of the serial dilutions were more than 116. Next slide. And this is a comparison of the initial and the follow-up questionnaires. And you see how they had gotten sicker during this period of time. Next slide, please. And the neurological symptoms were much more too. And the mental symptoms were more. And the ones that were on antibiotics, 7 were reported that they had more symptoms, but 6 reported they had fewer symptoms. What it means, I don't know. Next slide, please. Now, when I called these people, I did not talk to but one person. I talked with the nurses, I talked with husbands, I talked with sisters, and I talked with everybody except the patient themselves, because they were gravely ill and could not talk. And they still were carrying the diagnosis of ALS, and they had not been treated for Lyme disease, except one guy. I got him and I talked with him. He said that he had improved and he's now functioning as a human being. Next slide, please. Now, before this guy, I talked with him, he said that the Q-rib test inspired him to find a physician that would treat him for Lyme disease. But in trying to find somebody that would, he saw 6 neurologists at 2 internal medicine physicians who told him he had ALS and to forget about the Lyme disease. Quote, I'm quoting. So he finally found a Lyme specialist, and he's being treated properly, and he's improving steadily, and he says he has never felt so good in all his life, and that he owed his whole life to the Bowen test. Amen. Next one. Now we go to the validation of this test, and Dr. Mattman, who is here, she's the one that relieved my anxiety about this test, because we did the same draw specimen. She cultured out 613 Borrelia burgdorferi bugs, and we had the same positive test, no negatives. Next slide, please. This is Dr. Mattman's. This is a culture on an ALS patient. Shows a pleomorphic spirochetal colonies stained with a fluorescent Lyme antibody. Next slide, please. This is actually uncultured blood with a acridine orange stain, which just stains DNA and RNA. Dr. Mattman, I couldn't have given the talk without her slides. Next slide, please. This just shows that Dr. Mattman has found that the spirochetes of Lyme disease and ALS are similar in appearance with the electron microscope. Next slide, please. Now, Dr. Halperin, I don't know if he's in the audience or not, but he actually found that ALS patients had more seropositive for Lyme disease, had a higher percentage of seropositive for Lyme disease than other people, and that's the reference if you want to look it up. Next slide, please. And Dr. Hofer, a friend of Dr. Mattman, we sent him same blood, same draw specimens, 40 samples, and we did 40 at our laboratory. He got 40 positive, and we got 40 positive, and no negatives. Next slide, please. Now, Dr. Bruce Tudum, his daughter was very, very ill, and in fact she was dying, and they didn't know what was wrong. We did our test on her, found out she had Lyme disease, she was treated, and now she's well and going to college and doing well. Dr. Tudum was so impressed with the test, he came down to learn it, went back to where he came from to work with Dr. Stewart, and they did what I wanted Dr. Mattman to do many years ago. I wanted to get healthy blood donors. So I called up Lyda one day and I said, Lyda, I think we ought to do it on a healthy blood donor to show that they all had Lyme disease. And there was a big, long pause. And then all of a sudden she came back and she said, Joanne, she said, I want to live a few years longer. So that was the end of my testing healthy blood specimens. And thank you, Lyda, you probably saved our lives. Okay, now what they're doing is they get negatives, but they express their positives in a very unusual way, which I'll tell you about. But anyway, they were 82 These are healthy blood donors, 82 samples, 30 were positive, 19 were equivocal, and 33 were negative. But let me tell you how they did their positives and negatives. Next slide, please. You can see the positive, they had to have at least 4 or more fluorescing organisms. The equivocal were 2 to 3, and they were negative if you had 1 or less. Now, to me, if you have 1 fluorescing organism, I think you have to be very careful in saying it's negative. And actually, Dr. Stewart agrees with me, too. Next slide, please. We had Dr. Reisner. She did our test, and she also sent the Western blot test, which is an antibody test. And out of 57 specimens, Western blot missed it, 30 only found positive on 38 of the 57 patients had 19 negatives. But you can say we did agree with Western blot on 67%. Next slide, please. Dr. Bradford, who has a high-resolution microscope, he confirmed our test 100%. And we also have a doctor Dr. Andy Wright in London, who is using a high-resolution microscope, and he sees it in everybody. He thinks everybody has it too, like I do, and I think Dr. Mattman will tell you what she thinks. Next slide. Dr. Mattman suggests ALS is some form of Lyme disease, and these are classic spirochetes in the red blood cells. Next one. Dr. Koch explored the possibility that ALS is caused by a cell wall deficient organism. Next slide. Now, we rarely see the spirochetes when we do the Q-rib test. We see only cell wall deficient forms, which makes this organism a booger to treat. You see the fluorescent green color. Next slide, please. So, is amyotrophic lateral sclerosis, is it a late stage of Lyme disease? I think it is. Treated as Lyme disease can lead to a healthy, productive life. If not treated, usually death occurs 3 to 5 years after onset. If I have ALS, by golly, I want to be treated for Lyme disease. Thank you very much. I've enjoyed it.
All right, so I'm very proud to introduce Dr. Mattman, a pioneer in research in this field. Dr. Mattman.
I realize, can you hear me? That many of you, most of you have made a significant sacrifice of time and even a little money to be here. And in return, I have worked diligently, hoping this will be not only informative, but interesting. So here we go. As most of you know, my specialty is bacteria that float around in disguise, bacteria that don't look like themselves. Well, first we're going to talk about definitions and what cell wall deficiency consists of. So we see the diagram. We'll see all the great possibilities, and we're going to talk about why they are important, and of all the genera of microorganisms, spirochetes seem to be among the best at disguising themselves and not looking like spirochetes. In fact, all the textbooks say, don't try to ever see spirochetes. All you'll do is see fiber and debris. Just give up on it. But we know that isn't true. And we'll talk about a couple of diseases beside the spirochetal diseases, and 2 diseases in which etiology is solved by looking for pleomorphic stages. So it doesn't matter what it is to start out with. It could be a vibrio, a bacillus, a coccus, a spirochete, or it can be a Helicobacter. And Helicobacter is particularly interesting right now because Helicobacter pylori has recently been found to be a cause of gastric cancer. Probably not the only cause, but certainly that very important disease is caused in this case by an L form of Helicobacter.
Lydia, could you explain why the cell wall deficient form concepts are so, shall we say, backwater in conservative or traditional medicine? Because this particular slide would garner so much anger from the microbiologists, and I'm wondering if you could help us understand this great chasm between what you're putting up here and what we're taught in medical school about infectious pathology.
There's been a lot published about how long it takes before something is discovered and before it's accepted. And we talk about the brass monkeys who don't accept anything new, and the world is full of them. So that's what you run into, because it's too bad. In microbiology, they tend to teach what they could grow in an agar slant or petri dish, and what's so much more important and 10 times as fascinating is what's circulating in the blood and what's found in the body, whether it's a horse or whether it's man or your pet dog or whatever it is. What goes on there is so much more interesting and, of course, is what is important. Oh, wait, no, not yet. I'm sorry. Yeah, look at all this. We think cell wall deficient forms is not a very good term. It was given to them by a committee. A much better term, to think of CWD as standing for divergent, because this wall can be very different in diameter. These can be so little that they're filterable, like a virus, or they can be so big that in some instances they're seen macroscopically. So L form, we like the term L form because we can think of it as standing for little filterable ones, or so big that they're almost visible with the naked, with your usual microscope. But L forms was given to these because they were studied at Lister Institute. If you wonder where the term L form came from, it's much better. And we're going to talk about L forms and not cell wall deficient, which is harder to enunciate anyway. Well, all these are possibilities, and some genera prefer one or another. For instance, we're going to talk a lot about tuberculosis, and what we see there as the L form is like an actino. It has little filaments that branch, if you've ever worked with the actinomyces. So that's what the tubercle bacillus does as it grows in the body. It doesn't grow like it does in Petrogoni's medium at all. Very, very seldom you don't find it. It's in a different form in vivo. The next slide is of Almquist, who was in public health, and it was his hobby to look at bacteria growing atypically. And he said one lifetime, a whole lifetime, was too short to see all the variation that could be exhibited by a single species. And this is an example. You all know what E. coli looks like, just a little gram-negative rod. Who would know that this was E. coli? And this was antibiotic-induced. In the body, we feel they're induced by lysozyme and antibodies and natural immune phenomena. But of course, antibiotics are famous for inducing them. The next slide, this is E. coli colonies, the kind you could get from Escherichia coli. 3 different kinds of colonies. So we would be absolutely lost at identifying these if it weren't for fluorescent antibody. And I am proud to say that my mentor, Cora Downs, when she was at the University of Kansas, developed a method for making fluorescent antibody. Before that, they were afraid to make it because it took nitrogen mustard to make the combination. And she showed that it could be done with fluorescein isothiocyanate, which wasn't toxic. She said it only took them 1,000 hours, she and her graduate students, to learn how to make fluorescent antibody. But you see, we would be lost if we didn't have fluorescent antibody to identify these things for us, because their morphology isn't going to help. This is like asking a man to wear a tuxedo and a tie all the time. You know how men are dressed and what they prefer. They don't like a collar and tie. And bacteria the same way. They don't like to appear in their so-called classic stage. When we talked about the definition, it's important in livestock. It makes our meat bill very high. Benefits, well, you know that when food is broken down in soil, nitrogen goes into the air, and there are bacteria that are responsible for that. But you have to have something that gets nitrogen out of the air. So there are the very famous, well-known nitrogen-fixing bacteria, and these are all cell wall deficient. They're all variations. So none of us could be alive today. We wouldn't have any nitrogen for amino acids if it weren't for this cell wall deficiency phenomenon. And I put this just to be funny. It causes sterility in fruit flies. You want to get rid of those. Some people say it isn't limited to the fruit fly. We aren't sure about that. Well, these are diseases in which the L form of an organism has been isolated in pure culture. Uveitis, a perfectly terrible cause of blindness. I can show you horror pictures of those. Would you look at the fluid from the eye, the uveal fluid, what you find are L forms always. So there isn't a disease, and it's hard to find one that isn't mentioned here, okay? And these are the pathogens that have been isolated. They're fungi too, Candida. You find some more. Well, look at the mycobacteria and nocardia, and there's a second slide on this. The list is so long. Salmonella, the organism of typhoid. This is important in causing rheumatoid arthritis, believe it or not. Well, all of these can wreak havoc with the human body. Next slide. One problem that cell wall deficiency causes is that they find one organism in the classic stage and they think that's the answer. But whenever proper therapy for that organism doesn't work, they should very diligently look for a cell wall deficient form, an L form, also present. And this is a case where E. coli was with Staph aureus. This was in medium that was specifically designed to grow Staph aureus, a complete medium, but the growth as it starts out is not classic at all. It's cell wall deficient. When I first got into this, I wanted to learn from Dinas because he is the one who first showed how pathogenic these could be. He was in the Boston area, and I was at Wayne State. And I sent a student to him to learn about L forms. And I thought a couple of days. Oh, no, he wanted to keep her for weeks. There was a lot to learn. And so those of you who are teaching, there is quite a little bit to learn here. Well, this is what happens when you don't have an early diagnosis of staph. This was an infected mother, and the infant was infected, this little boy was infected as he was delivered and died by 5 days. This is plain Staph aureus, but the diagnosis looking for the L form could have been made very early. This one I put in case you haven't had enough coffee yet today. This would wake anybody up. This is cutaneous TB, and the diagnosis was greatly delayed, and this woman's face was completely mutilated before the diagnosis was made. The organism from, by the time they finally made it, it was over a month. Anyway, let me say that fortunately she found a plastic surgeon who was able to restore her with a normal face, but of course not at all the way she had originally looked. And this one is the most pathetic slide I have, I think, because this young man died 5 days after this was photographed. Addison's disease is tuberculosis of the adrenals, and he had pulmonary and adrenal tuberculosis. The point is here that if they had done a blood culture and used even a simple method that we use, by 48 hours for sure, usually at 24 hours, they would have grown the L form of the tubercle bacillus. And it's very easy to stain using the stain that's in my book. I have a few here for sale. And it's intensified kidney stain will identify these as acid-fast, and this patient's blood was loaded with it, I'm sure. So a blood culture, not any special medium, the medium that's commonly used for blood culture in this country or Europe would grow this usually in 24 hours. So you'd have a 24-hour diagnosis, at the latest a 48-hour. Russia is always getting ahead of us, you know. They got to the North Pole first by submarine. They've done all kinds of things ahead of us. And this is another example. Maybe they have something better than we do, because in the TB SAN in Moscow is a woman investigator who has a test that is 85% positive the first day. Just take the blood out of the finger or the vein, and with her special technique, you can recognize the tubercle bacillus in 85% of the cases. Well, we get it in 100%, but anyway, I think, watch the news. Sometimes she's going to have her test in a form that can be used by other people. So far, she hasn't been able to help us. She's been trying to be cooperative, but you know, things can be seen under the microscope that can't be photographed, and the photographs she has sent us are not useful at all. But if you watch the Russian literature, maybe you're going to have a test that is positive as soon as the blood is taken out of the vein and stained. It's a special stain, too, not one you can buy, one she makes up. So you have your choice of the Russian method, which isn't complete yet, or you can have a 24-hour method. Our method has been adopted in China, and it saved the lives of many, many people there. And it saved the lives of many people in the ghetto in Detroit, where there was a lot of tuberculosis in the black population. And we had the pleasure of seeing these people walk out instead of being carried out with a sheet over their face, because diagnosis of tuberculosis is easy if you do it right and you do it early. See, this is what happens to the lung when the diagnosis isn't early. The lung isn't full of air sacs, it's full of hemorrhage and debris. The culture was positive only at 8 weeks, and it led to this. It was something my students taught me, you know, Abraham Lincoln said, "All that I am or hope to be, I owe to my angel mother." Well, maybe all that I am, I owe to my graduate students, because when you've got a dozen graduate students and they're all smart, you learn a lot. And that was my fortunate situation. Well, this is the most valuable photograph I have at all. This is the way the tubercle bacillus, unfortunately, this colony got torn a little bit, but this is the edge of it, you see, does look sort of actinomyces-like. There are filaments that branch. And this is what you'd find in a 24 to 48-hour blood culture of any patient with tuberculosis in any part of their body. It can be on the skin, it can be the adrenals, it can be the lung, it can be the genitals, any site. It'll get into the blood and can be cultured from that. So you can do this with fluorescent antibody too, of course, but not everybody has a fluorescent scope, and it's hard to keep fluorescent antibody on hand for all the organisms that are encountered in the clinical laboratory. Well, this is a different subject, and this is the first time this slide has been shown to a group. I think this is a pretty exciting subject, and one for the future, surely, unfortunately. This is a colony from a mouse given the L form in the pneumococcus, and the animal developed myocarditis. That's inflammation of the cardiac muscle, of course, and it's very common, and the cause is unknown, and it looks as though it could be something as common as the pneumococcal L form. Well, the point of all this is this could be prevented by vaccination, probably. So maybe this should be added to the vaccines that are given to everybody, to all children, because a terrifically high percentage of people develop myocarditis, and the cause of it has been unknown. As something was said by Joanne a while ago, acridine orange stains nucleic acids. So if you have a staining of anything with acridine orange, you know it has DNA or RNA in it. If it's orange, it means they're actually multiplying still. It's RNA. If it's green, it's DNA. They've quit active multiplication, but you still have DNA there. Anyway, the point is that this had living cells in this colony. Isn't this perfectly horrible? It says in the absence of a positive blood culture. This is a man with Pseudomonas, and the blood culture would be positive for L's, I'm sure. They wouldn't look like pneumococci, but like Pseudomonas, but they could be identified with Pseudomonas antibody, even using the direct method, the indirect method of. There is another horror story of 16 cases of this Pseudomonas infection in people who had shared the same spa, who had been to a health spa and been in the same bath. So these are what you call water bugs. They could grow in nothing but water. And people say, well, everybody knows bacteria can't grow in just water. And on the contrary, there are some very important pathogens that in the L phase grow in water, and Pseudomonas, unfortunately, is one of them. This is water which was sterile by putting it on the surface of an agar plate or putting it into so-called nutrient broth, just plain nutrient broth without any yeast in it, no growth at all. But you make a pour plate, and it's full of L colonies of Pseudomonas aeruginosa, and these were stained with just carbofucsin. Well, then I don't know whether this is a topic that belongs in here or not, but I put it in with the point that it could save your life. To know this might be the difference to somebody here between living and dying, because this concerns the very common disease, coronary thrombosis, the heart attack, the sudden piercing, terrible pain. And it's not amusing, but I don't want to make fun of a country, but there's one country that before the cause of this was known, thought it was a crisis of the liver. And so they were desperate to find out what caused this terrific pain in the chest, and they couldn't find any cause. They did autopsy after autopsy and couldn't find any reason. And then a professor in medicine at Johns Hopkins died, and this was one of their own, and they did a very careful autopsy, and they found the clot in the coronary artery. So they know it results from obstruction of blood going back to the heart. And my husband had a coronary, and this is how I got involved in this. They all develop, every case of coronary thrombosis develops fluid in their pleura or their peritoneal cavity. So they have either pleural fluid or pericardial, I'm sorry. Either, and so this happened to be pleural fluid anyway. In the pleural fluid from every case, we found there was a pathogen. And it wasn't any bacterium we'd ever heard of. It wasn't an L form. It wasn't a mycoplasma. It wasn't a fungus. The virologist looked and looked and looked and could find no virus. So what was causing the clot and this fatal disease? Finally, it was solved by someone in Scandinavia, and it is chlamydia pneumoniae. We found it would kill guinea pigs, and we could grow it, but we couldn't name it. But anyway, it did get named. Well, the point of this that applies to everyone here is that if you have a friend who suffers a coronary, talk to him very pleasantly on the phone and send him flowers, but don't sit by his bedside. We think it's contagious, and the incubation period is 4.5 months, which makes it hard to trace. Furthermore, a woman who takes care of her father who has the coronary, in 4.5 months, she has good arteries, so this doesn't cause an occlusion in her, but she can be infectious, and she can give it to her husband, who will have a coronary 9 months after her father's. So the way you could help on this is if you collect anecdotes, send them to the people who are interested in this. We have a few, like the man who developed a coronary 4.5 months after he was hospitalized in the same room with the coronary, and 2 men who were carried out in stretchers who sorted mail in a little mail room, and supposedly 4.5 months previously, they had sorted mail with somebody who had become an active, about to roll coronary case. You can say this is cell wall deficient because if you look at the electron micrograph of chlamydia, it has a very funny wall. Well, this will wake you up too. This is a spirochetal disease, and so we get to spirochetal diseases. And this is due to infection of the soft palate and part of the nasal structures, and is caused only by yaws. My point here is that spirochetal diseases tend to become very widespread and finally endemic. We think that has happened with Lyme. In the 1960s, a long time ago, in Haiti, yaws became endemic. Everybody had it practically, many, many cases. And they didn't say, come into our public health laboratories or offices and get therapy. They went to the houses and they shot everybody with penicillin. And this spirochete responds to penicillin. But the trouble is, the spirochetes we're dealing with Lyme, etc., are not sensitive to penicillin. We don't have a magic bullet. Anyway, this is one that is called as a famous spirochetal disease. This has been with us since man was first in America, apparently. It's been found in the graves. Okay. One of the most fascinating things about these spirochetes is that they think they're a protozoa and they like to dive into red cells and live there like a malarial parasite would. And this is ear prick. So if you have a cooperative patient who lets you use their ear, for some unknown reason, it's a little better than the finger. The finger helps too. But if you have venous blood or finger prick blood or ear prick blood, it's amazing how much you can find in a direct smear. The first time I saw this phenomenon, it was blood from Scandinavia, and I saw red cells that were just loaded, and I sent back the report that there were a lot of spirochetes and outside and many inside the red cells, and I said this couldn't possibly be a direct smear. Well, I got a call from Dr. Berg in Stockholm, and he said in his perfect English, I made those smears myself. Those were direct smears. And we found it was true, that people would be walking into our lab. They didn't feel well, but they didn't feel perfectly lousy either. They were able to teach or function fairly well, and their blood would be so loaded that you didn't even have to culture it. You could just take a direct smear and look at it. So this is something that anybody with a microscope can work with. This is some more parasitism. This is 5 days. The ideal temperature is around 35, but 37 will do, of course. And one thing to notice is that people tend to look at the thermometer at the top of the incubator and think that's the shelf temperature. You want to keep a thermometer on the shelf and know what the shelf temperature is. Carbofuchsin is an excellent stain, and this shows it can either stain or it can stain them negatively. So either way, carbofuchsin is the best simple stain for this. You notice the magnification is 1500. We use 1.5. Well, most people are satisfied with 1,000, but this is a little more. Okay. And some more parasitism. See, by the time blood gets to you, unless this is somebody who walks in the lab, it's been shipped in the mail and the temperature is adequate, usually on the plane, and so that you get growth occurring in shipping. Well, something very unexpected and interesting and helpful is that often the spirochetes are autofluorescent. So this isn't stained. All you need to do is turn on your UV of your microscope, and you can see these autofluorescent spirochetes. And again, this is just shipped blood. And this will bring us to a point that I can't emphasize too much, that you don't need to worry an awful lot about culture medium, because the best culture medium, an excellent one, is the patient's blood. And they like it just fine. So you can incubate it, and even the refrigerator, they continue to grow as long as they're in blood. This is a fluorescent Lyme antibody, and this is what you expect to see in a stained smear. This is a 4-day blood culture. And then we get to xylol concentrate. After you've incubated blood, the most sensitive way to determine whether anything has grown in it is to make a xylol of concentrate of it. So say you take out 1 ml of the blood that's been incubated, and you put in 1/10 of an ml of xylol, and you shake it for a minute, and then you let it stand at an angle, and you'll have a little corner there where the xylol will come to the top, and you can extract the xylol. The point is that these bacteria have a lot of lipid in their walls, and they tend to go into the xylol instead of staying in the blood plasma. So in that drop of xylol, you'll have a concentrate of everything that was in your whole ml of blood that you're studying. And this was a gorgeous L body, and with spirochetes growing out of it. Well, something that isn't well known at all, I think, is that this is a congenital disease and a venereal disease, and we've only studied a few samples of semen, but this was in men who were suspect because their wives and their children had Lyme disease, but it's easy to grow spirochetes from the semen. This is the work of Alan MacDonald, who has published quite a bit on this. It isn't syphilis that they fear now as being carried congenitally. It's Lyme disease. And this is showing that the fetal kidney can be infected. See, these are autopsies on dead newborns, and out of 24, 4 had Lyme infection. So that's 1 out of every 6. Well, that's quite a lot. Well, this is some cheerful news for anyone who's in a laboratory and you don't have a fluorescent microscope and you haven't bought fluorescent antibody. There is still a simple test you could do that is a presumptive test for spirochetes. And this is Sudan black B. Some of you may know it's used to diagnose Pseudomonas. Well, how many patients have Pseudomonas? Not very many. So if you stayed with Sudan black B and you have something like this, you have a presumptive diagnosis for Lyme. And again, this is direct smear. This is acridine orange again, and this is in urine, a pleomorphic colony with spirochetes. And this is acridine orange again, a xylol float, 3-day blood culture. So acridine orange is great help. And this is another acridine orange showing that every red cell here has been parasitized. This is a Lyme patient, his blood incubated for 10 days. And then we get to the gist of all this, and you're one of the first audiences to hear this. Well, anyway, first we're going to talk about things that go along with the spirochete and electron microscopy. The EM was done by Carol Ayala at the Core Research Group in Providence, Rhode Island. I think she is one of the finest electron microscopists in the world. Other people can just do nothing for us. But she and one other lab we know of know what they're doing. Anyway, this is what you see if you do electron microscopy on a blood culture of a Lyme patient. One thing we have done, we have developed a medium that has grown spirochetes in 500 cases, and we have a good medium, and I have it in black and white. I can hand it out to any of you. I cried a few months ago, or some time ago. A paper was published by a very outstanding laboratory in the United States, and they said our medium wasn't any good. They couldn't grow spirochetes. They tried, and they looked, and they waited, and they looked. Well, why didn't they call us? There are 10 mistakes you could make, and the question is, how many of the 10 did they make? And there's nothing easier in this world than not growing spirochetes, and yet some journal was willing to publish that. Anyway, we do have a good medium. It took us 5 years to develop it, but it'll grow it from the blood. BSK medium grows it from spinal fluid, but who wants a spinal tap? It gives you a headache the next day. And people are a lot more cooperative about giving their blood. Anyway, these are the cysts that accompany the spirochete, which we don't show here. But these are characteristic cysts that are found in Lyme disease. It's either this or the next one. These are different genotypes, of course. These are attached. These little things, I used to think they were flagella, but the flagella experts say no, they're not the right size, so we can call them fimbriae, which means a hair-like structure. So here over here someplace you have big spirochetes, but these are the cysts. The Germans call these cysts, these round, most cases round form that go with the spirochete. Cyst is a pretty good name for them. So these are spirochetes in Lyme, and they aren't like anything else, like the others. Go quickly through this. This is the evidence. Well, not that fast. Anyway, these are the evidence that multiple sclerosis is due to a Lyme, to a spirochete. And these men in Europe have found a spirochete in spinal fluid and done Koch's postulates. They've injected animals, and the animals have gotten sick, and they have found the spirochete again. And that's Koch's postulates. So Gabriel Steiner in the United States, he was at Wayne State, incidentally, he found it in the MS brain, an autopsy, again and again. And he said it's not treponema, it's not syphilis, it's more like Borrelia in its shape, morphology. And 50 years ago, he was right. But anyway, Steiner first published in Germany, and then he moved to the United States. But, okay. And this is what you find in spinal fluid of MS. You don't even have to centrifuge it. Of course, you get more of them if you centrifuge it. Well, this is work of Brorson, B-R-O-S-O-N, in Norway. They've found that in multiple sclerosis, in addition to the spirochete, which would be out here somewhere, you have the cystic form is characteristic for multiple sclerosis. It has what I call backpacks on the cyst. And they did a series of multiple sclerosis cases looking at the spinal fluid itself and looking at cultures in BSK medium. And in every case of multiple sclerosis, they found this characteristic cyst with backpacks. But the important point is we find the same thing in America. So this, even the brass monkeys might accept this. I hope they will, that a spirochete is involved and it has the characteristic cyst. Okay, the next one. This is multiple sclerosis again, and these flagella, we call them here, you should call them fimbriae, I guess. They have swellings in the, and it's only in MS, it's not in Lyme, it's not in ALS, it's not in Parkinson's, it's only in MS that you have these swellings on the fimbriae. The next one, please. And this would go to Lou Gehrig's disease, ALS, and the cysts, again, are different, and they're only like this in ALS. And here again, the fimbriae are unique, they're segmented, interrupted. And all 4 of our ALS patients had this, and it's the same in Europe as it is here. We were doing work for 64 doctors. We had plenty of samples to look at. Here is segmented, enlarged more, okay? This is Parkinson's, and this is really new. I know one physician who thought Parkinson's was just a manifestation of particular susceptibility on some patients, that it was really Lyme disease. So he treated 5 Parkinson's patients vigorously for Lyme disease, and they all improved. And I know of another physician who did the same thing with one case. So Parkinson's is not only the etiology is solved, but the cure, the therapy is also there. This was with immunogold, which is considered better than fluorescent antibody for some reason. There are little black spots in here. Immunogold antibody, just to really be sure. And this is the second kind of cyst you see in Parkinson's, and immunogold is confirmed that it is reacting. The point is that all of these are Borrelia. They all react with Borrelia antibody. They're in the Borrelia genus. It's like Staph aureus and albus. These are Borrelia parkinsonii, or whatever they want to call it, and this is the one of Parkinson's. This is negative staining with carbofucin, and some more negative staining. Just showing what you can do with a very simple stain, more negative staining. I've got enough for 3 hours. This is one you saw before. This is just showing that all of these from MS or ALS, they all react with Borrelia burgdorferi antibody, but the cysts are different, so we know they're 3 distinct diseases, okay? This is just blood that was long refrigerated, and you get a fluorescent antibody reaction of pleomorphic stuff and of the spirochetes too. This is Borrelia burgdorferi antibody, okay? Oh, this is Lou Gehrig patients. I just thought this was interesting. This is a 10-day blood culture, and the colonies still stain red, showing that vigorous multiplication is occurring, and that isn't true of MS and Lyme. It's ALS. It's a deadly one, which is so awfully sick. This is the last slide. Okay.
Thank you. I know you're probably all restless and tired, but I wanted to hear from clinicians who have been dealing with this for years and years and years, who have been fighting with the rest of the medical establishment to try and treat the patients that we know do respond, and I'm very proud to introduce Dr. Bernie Ratzler. Bernie.
Well, today I'm going to try to go over a couple of topics, but I frantically changed my talk today because I got the April 2005 Psychiatric Annals, which goes out to most of the psychiatry world in the United States. This is a throwaway journal, but it's an easy-to-read one, and it deals with a different topic every month. And this particular topic is called Medically Unexplained Symptoms. So let's learn something new about psychiatry. First slide. Oh, he went, yes, he'll be coming right back. So MUPS, which is called Medically Unexplained Physical Symptoms, these are complaints of physical symptoms or signs for which there's no adequate objective psychophysiological evidence to explain the distress. Next. Now, they're encountered in medical settings where patients and clinicians are often unfamiliar with psychological explanations. Next. By the way, this is condensed from the lead article, so these aren't my words. Now, it implies the presence of symptoms that do not conform to known disease processes. It defines a predicament rather than a disorder, a way of drawing attention to societal situations in which the meaning of distress is contested. It is critical to accept that unexplained doesn't necessarily imply purely psychological origins, as the history of psychiatry, as we know, is replete with examples of disorders that once upon were functional, but then we discovered there were certainly proven to be organic processes, and the classic model, of course, is the dementia and the whole plethora of symptoms caused by syphilis. Next. Some patients may have vague symptoms initially that appear to be unexplained, only to manifest clear evidence of the disease later in the course of the illness, like MS or myasthenia gravis. Next. So let's go to the epidemiology. Why is this happening according to this article? Next. No, we're going to pass that one that just tells us that the economic costs exceed $100 billion for MUPS, and it doesn't include substantial indirect costs of lost workplace productivity and disability payments. Next. So
Now you break down MUPS, or medically unexplained symptoms, into specific somatoform disorders. Next. Here they are. So, it can be delusional disorder, somatic type. It can be a somatization disorder, a conversion disorder, hypochondriasis, body dysmorphic disorder, pain disorder, facetious disorder, or malingering. Oh, those are very good ones. We're going to find a lot of this in this room, I'll tell you that right now. Next. Now, this is a psychiatric explanation, early one. Psychodynamic hypotheses were based on studies of hysteria. And central to this line of reasoning is the notion that conversion reflects defense mechanisms that symbolically transform unconscious emotional trauma and conflict into physical symptoms. So, you got a swollen knee the size of a melon, and you got an arm you can't move to your ear. You got some problem, because if we work you up and you don't find nothing, you have an unconscious emotional trauma, and that conflict is being, yes, something to do with your mother or stealing watermelons when you were a kid, something. Next slide. Now, we threw that one all out. I'm proud to say, a psychiatrist. And now, if you understand this, you could take my place at the podium. Here's the explanation for MUPS. It's the activation of or regression to subsymbolic somatic symptoms dissociated from verbal symbolic representations of feelings and objects. Patients therefore experience somatic signs only, detached from psychological meaning, or in other words, they exhibit alexithymia. This goes out to the world. Now, what does that mean? Can anybody tell me what that means? Because I don't know what that means. Okay. Now, I am going to show you some MUPS. You all ready for MUPS? Because these are the cases that came into the practice. 34-year-old male comes to the office complaining of headaches, weakness, stiff neck, fatigue. He has increasing difficulty in the fall, staying focused. He never feels rested. He always feels depressed. In March, he develops severe dizziness, lightheadedness. He becomes sound sensitive, dizzy. He passes out. He goes into a seizure. He gets put in the hospital. He gets dismissed as an anxiety attack. June 2004, the migraines become unbearable. Total body weakness, stiff neck, oval rash, negative EEG, MRI, CAT scan, spinal taps, all negative. Finally, someone in Maine got smart, did an ELISA, and he had a Lyme confirmation of 4.7. Diagnosis, somatoform disorder, pseudo-seizures, and malignant depression. Next case. 42-year-old female presents with persistent sinus and sleep problems, TMJ, locked jaw, feeling lightheaded and dizzy, not related to any activities. Now she starts to have cognitive problems, including concentration, word retrieval, more depression, frequent headaches, nightmares, night sweats, chills, extreme fatigue, poor stamina during the day. Completely worked up, find nothing, got to call it MUPS. So I would say she has a somatoform disorder with malingering and hypochondriasis. Next patient. So we got a 43-year-old female who presented with the following symptoms: migrating joint pain, fatigue, memory problems, on and on, feeling suddenly overwhelmed, drenching night sweat, short-tempered, and enough to keep her out of work. Someone had her on Plaquenil for her joint pain and prednisone for 3 months. I would call that one what would I call that one? I would probably have to say that that would be a facetious disorder. That's pretty good under that classification. Because how could she have all that? I mean, clearly every system in the body is being tagged, and we found nothing, quote, so it's got to be facetious disorder. Next case. This is another woman who had muscle weakness, soreness, photosensitivity, sweats, decreased exercise tolerance, decreased energy, fatigue, balance difficulties, all these physical symptoms, and reports also trouble with her memory, difficult thinking, feeling confused and easily distracted, no diagnosis. Pick anyone you want from MUPS. Next patient. Now, this is an interesting one. I'm going to read this right to the end. He's a little guy, 10 years old. He presents complaining of bugs. We like bugs, crawling all over his body. He has developed an increasing fear of separating on death, so he would never leave his mother's side. He had a sudden onset in February. He became more and more afraid and agitated. As weeks went on, he felt he was going to disappear. His room was going to catch fire. He felt the ground was going to fall beneath him, refused to be touched, kept describing the sensations of bugs crawling all over him. He saw a psychopharmacologist, child psychiatrist, who put him on Risperdal. Doesn't say here, but he was hospitalized for 6 weeks in a child psychiatric unit. He was completely worked up, including EEGs, spinal taps, MRIs. Everything came back normal. He was discharged home. He was homebound. He was school tutored and continued to have the delusional system of the bugs crawling over him. The diagnosis is childhood schizoaffective disease with severe anxiety over separation, individuation, and body dysmorphic disorder. Okay, to the lab. Next slide. So what happened to MUPS? So let's go back to the first case, see what happened with different lab tests. Next slide. Well, how do you like that? IGeneX showed a Western blot of IgG positive, IgM positive. Babesia and auricular titers were negative. Mycoplasma was negative. But the SPECT scan, this as read by the radiologist, showed subtle hypoperfusion, and it involved a lot of the brain, right posterior parietal cortex, basal ganglia, which is very important, particularly in the light of this presentation. Highly suggestive in an appropriate clinical context to be compatible with mild Lyme encephalopathy. And I'm going to go a little bit more into how the presentation of encephalopathy can be so confusing. But here we went from medically unexplained psychiatric symptoms or disorder into a very clear picture. Now, what's important is that each of those cases have a similar dynamic, and that dynamic is this whole general system presentation. Anyone who treats Lyme, when they hear that someone has insomnia, or they have a short-term memory problem, or they suddenly can't remember names, or they're a little confused when they're going to the grocery store, or they forget their lists, or they're putting post-its on their forehead so they don't forget to pick up the list, which is to remember what groceries to get and how to get there, is a red flag. And that gets into this whole question of the subjective symptomatology in this case and the prejudice against it, and I want to get to that in a second. Next case that we saw, the second one that I read, positive Western blot and a positive Babesia, possibly potential Bartonella. We repeated it, and in fact, it did come back positive. And the SPECT scan showed lots of pathology, including a decreased cortical uptake, temporal lobes, frontal lobes, and an area in the right frontal parietal lobe. Now, you know that the frontal lobes are the lobes in which you master your general day-to-day tasks. It's the place you take short-term memory and you use it to complete a set of tasks. It's also actually involved in irritability. It's involved in a number of other processes like functions in space, and some such. You can't read, you can't concentrate. You see a lot of SPECT scans showing the frontal lobes being hypoperfused. Next. This patient, again, was also diagnosed with Borrelia and Babesia, and that she had positive Western blots, positive immunosciences, Babesia and Bartonella peptides, and a positive SPECT scan. Next. Again, another whole set of positive lab studies, and we'll go to the next. And finally, the little boy, he showed exposure to bands 45 and 58, and an IgG of 41 and 66. No oviparasites were found. He was strep negative. The one little clue that I had on this youngster was that he used to vacation, he was from Brooklyn, and he vacationed on Cape Cod, which kind of rang a lot of bells. And so we treated him very simply with Biaxin and Ceftin, and within 2 months his symptoms had completely gone into remission. No more bugs. In other words, no more peripheral neuropathy. He was quite right. There were, in fact, bugs crawling on him. Next. Now, I'm going to, how much time I got? I got 15 more minutes. Okay. What I want to do now, with your permission, is I usually speak extemporaneously, but I spent some time writing some stuff, so one thing I really hate is to be read to. I'll be sitting reading my section of the New York Times sports page. My wife will be on the editorial page. She'll say, honey, you got to hear this. So I get read to. My father used to read to me, and I didn't like it. And so now you're going to have to suffer the indignities of being read to. So can I have the next slide, please? Next one. Yes. So, having seen the way these symptoms can sway across the borders of all the different subsystems and specialties, I believe that we have a new medical subspecialty has been born. I've come to understand that the treatment of the tick-borne disease has the potential to become a new medical subspecialty. The tick-borne disease physician must be prepared to deal effectively across many specialties and courageously cross taboo boundaries that define these specialties: rheumatology, neurology, psychiatry, pediatrics, infectious disease, and endocrinology. In too many instances, the medical community at large has often taken a skeptical and dismissive approach to this medical conundrum, with the unfortunate result that many tick-borne diseases are being missed or misdiagnosed. I don't call it Lyme disease anymore. I think, unfortunately, even though all of us have that as part and parcel of our vocabulary, I think the way maybe to get in part to the medical community at large, because Lyme disease is now so loaded affectively with, Lyme rights movements and all you folks bringing data to the doctors just to get them to see about this Lyme disease. I mean, it's now, the word has become almost a, a prod or like electric shock to the doctor. When I talk to patients, I say, for God's sakes, you're gonna get a consult, just don't mention Lyme disease. Don't say Lyme disease. Don't bring them all that stuff. Don't bring them Joe Burrascano's 41 pages. They're all right, of course, but don't do that, because Lyme rings these bells in the medical community. They're very defensive. It's not the best word to say. So I think we should shift it to tick-borne diseases. Now, that's okay. Tick-borne diseases, they're all over the world. You can get in line with that. If you've got deer running across your property, it's not too hard to think that there's a tick. And it's not too hard to understand that they bring bad things, They're sewers, so tick-borne disease. It's nothing about Lyme. Who, me? No, it's just tick-borne disease. Okay, next slide, please. So I asked the question, well, why does it seem to be that this particular illness provokes such a controversy in the medical field. I remember talking at a conference with Dr. Leslie Fine almost 15 years ago. It really dates us, but I don't think, in some ways, that the whole medical profession has moved very far in that time. I mean, Dr. Fine was seeing this and treating this aggressively in New Jersey 5 years before I even came onto the field. So that's 20 years in the field for her. And I was just talking to her outside, and I said, do you think there's been much movement? And, well, she'll tell you her own opinion down the road. But unfortunately, 20 years have gone by, and I haven't seen a whole lot of change. I think there's more an awareness, but I don't see a whole lot of change in the treatment component of this disease. We have a problem in medicine, and it arises from the physical problem of the mind-body dualism. And this is better recognized as the infamous split between our subjective consciousness and the material reality of our physical body. Unfortunately, the concept of mind or consciousness has been treated independent of our physical bodies. Modern medicine has practically ignored the critical relation of the metaphysical mind to the physical body. Instead, it has concentrated mostly on the physical brain as it relates to its physical body. In fact, it seems that many physicians do not welcome mind or subjective symptoms. Mind only becomes important when it is recognized as an extension of our hardwired neurobiology and can be treated with modern psychopharmacology. This mysterious vascularized gray and white mass of spongy synapses is the undiscovered territory of neuroscience. I took a long time to write that sentence, by the way. I like that. I'm going to make it listen. Sophisticated neuroimaging techniques, MRI, SPECT scan, and others, biochemical neurotransmitter analysis, genetic investigations of gene-driven mental illness, brain peptide production, single-cell evoked potential probes, and microscopic neurosurgery have allowed researchers to better understand the brain's previously inaccessible neurological secrets. The results have been spectacular. We have learned more about the brain and its workings in the last decade than all the accumulated knowledge of 100 years. So why the lag, then, in the clinician's office to what we, and many of us in this room, both have suffered and have attempted to treat, what has been put aside as the mind symptoms, the subjective symptoms? So let's take a look at how a patient might think. The patient's symptoms of tick-borne disease often appear in a disconcertingly vague and unclear manner. And for that reason, they're unconvincing to the inexperienced clinician. And of course, there are just so many ways that tick-borne disease can present, just like those 4 cases. Patients describe various degrees and intensities of pain. Sometimes it's there, sometimes it's not. Migraine-like intense headaches, peripheral neuropathies, such as this youngster, burning and stabbing and hot and cold. Sudden, inexplicable neurocognitive discrepancies, blunted or exaggerated emotional responses to the happenings of their daily life, depleted energy level, experience of overwhelming fatigue, extreme personality changes, including loss of libido, anxiety, depression, paranoid thinking, sudden OCD traits. Every single symptom that I'm naming here, I can give you 4 cases, 5 cases, 8 cases that presented just the way I've just said. Chronic sleep deficits in spite of extreme exhaustion. Sleep apnea, lots of sleep apnea. Persistent short-term memory difficulties. Focus and concentration problems, especially with reading, writing, and speaking. Word search and word substitution. Nonsense words approximating the actual word sought. World in which you live has changed dramatically, has changed in a way that you no longer recognize self. It is a disease of loss of self. And I, again, can't tell you the 3,000 plus patients I've seen where the patient will come to me and say, "Doctor, I want myself back." Now, lastly, there's a double rejection of the tick-borne patient who has psychiatric symptoms. These are a special class of the walking wounded. They are unsuspecting victims of their undiagnosed invisible disability. Their quality of life has been turned upside down. Their sense of self and their belief in their ability to distinguish psychological symptoms from physical symptoms is severely compromised by continual negative medical feedback. These individuals feel demoralized and are often left without hope and sometimes even without family support. And because of their general frustration with the medical profession, these patients, based on their own negative life experience, end up being insistent, demanding, and difficult people to treat. They push their doctor for an explanation of all the medical information they have researched. Some of the information is obviously of importance, obtained from journals or published articles in popular magazines. Some of the information is derived from association with other patients in self-help groups and tick-borne disease anecdotes. And most of the time, the information is gleaned from the wonderful, ubiquitous internet. Large percentage of informations, volumes and volumes and volumes, wheeled into the doctor's office, brought to the physician by the overwhelmed and frustrated patient, tends to be somewhat exaggerated or inaccurate. But an unyielding patient can produce legitimate physician frustration when they insist that every symptom is directly related to Lyme disease. And they will cling with absolute certainty to the belief that they are unshakably correct and will entertain no other diagnosis for their medical condition. And more times than not, they are right. So, finally, the psychiatrist's role in tick-borne disease, not calling it MUPS. The term empathy, which is supposedly the psychiatrist's main suit, his main tool, comes from German Einfühlung. It took a while to get that one out, which literally means in feeling or feeling into. The precondition for the empathic dialogue is a searching attitude by the doctor of simultaneously knowing and not knowing. Psychiatrists, with their training and supposed listening skills, their comfort level with the importance of subjective symptoms, their longer sessions, their understanding of stress symptoms and situations as it affects the immune and the neuroendocrine system, their emphasis on complex psychological connectedness, their understanding of the evolution of disease over time, all of these attributes make them an excellent physician advocate for diagnosis and treatment of tick-borne disease if they could only be taught to be aware of its existence as a diagnostic possibility and to recognize it in their psychiatric practices. With this, I'm going to end because I want to keep it within the half hour as I had promised. And just to make one overall comment, the two magnificent ladies that you heard first who've devoted their life to the study, research, and refinement of the technique of diagnosis of microbiology, really, and besides Lyme disease, these are the shoulders on which we clinicians stand. Because ultimately, it has to be an issue of science. Ultimately, it has to be an issue, some way of proving that everything that we talked about here and everything that we saw up here can be coalesced in such a way that the clinical picture can be recognized and that we at the other end can have something as clinicians to say besides our intuitive and our experience, which is valuable and critical, but we do need to have diagnostic procedures that scientists like Dr. Mattman and Dr. Whitaker have spent their lives attempting to develop. If you see the slides and you hear what they had said, there's so much in that hour, for example, that Dr. Mattman presented on ALS and on all these conditions. I mean, it would shift the ground. It'd be like a tsunami in medicine. So we're hoping that the information that is gleaned and put forward in conferences like this by women like Dr. Mattman and Dr. Whitaker somehow become an enzyme for change. So we clinicians who are in the trenches have more than our intuitive and our clinical judgments to say to the rest of the medical world who we want to convert. We're not against them. We just want to say, hey, look what we're seeing, guys. It's your specialty. Go ahead. I mean, that's where it's sitting. That would help us all. So with that, thank you for your time, and look forward to talking.
I think he deserves a standing ovation, actually. That's one of the best talks I've ever heard. That was the most eloquently, most beautifully described emotions that I've been feeling for the past 20 years. Thank you. I wish the research would be the tsunami, but honestly, I think that you people have to carry the torch. Because despite this research, the CDC puts out a warning a month ago saying that they are warning doctors because other doctors are using inappropriate testing for Lyme to make inappropriate diagnoses based on these tests. And there's nothing we can do on our own. There's not enough of us to petition and to lobby against this kind of thing. So we need your help.